Today we'll be talking about two different methods for gene editing, RNAi and the CRISPR method. Starting with RNAi, it is known as an RNA interference, which shuts off gene expression using double-stranded RNA. It begins with the dsRNA, which is cleaved by the enzyme dicer and forms mRNA or siRNA. These then bind to proteins to form an RISC, also known as RNA-induced silencing complex. They then base pairs with mRNA and either inhibits translation if it's miRNA or degrades mRNA if it's siRNA, and is used more for eukaryotic cells. The next method is CRISPR, which is an acronym for Clustered Regularly Interspaced Palindromic Repeats. It was adapted from naturally occurring genome editing system in bacteria, prokaryotes. This system is similar to the one that is used in the lab. Scientists will begin with a small piece of guide RNA that binds to a target sequence of DNA. RNA also binds to the Cas9 enzyme. The enzyme then cuts the DNA at the targeted location. Scientists then insert a new DNA sequence. Now, which method is the better one? Running through the pros and cons of each one, the pros for the CRISPR method is that it's often used in cancer therapeutics, curing genetic diseases, drug research, and it silences genes permanently. While the cons for the CRISPR method is that it changes to germline cells, chances of causing mutations, and has a high cost. While the pros of RNAi method is low cost, it helps study gene function. It's also used for medicine, like therapy, and has uses in agriculture. The cons of RNAi method is that it silences genes temporarily, and there's also a chance of causing mutations. So between the two methods, I would have to go for the CRISPR method because it is longer lasting. And here are my resources.